there are trillions upon trillions of viral variants. And a very, very tiny fraction of those pose a human risk, almost an infinitesimally small fraction. And so the vast majority of viruses are harmless. And then the theory would be, well, there are some that are still harmful, and we need to understand those, and fair enough. And one of the ways of understanding them is to produce more dangerous viral variants in the lab, and then we can study these more dangerous variants. But that seems to me to assume that you're presuming that the more dangerous functions that you're adding to the viruses are in some sense going to be representative of more dangerous viruses in general, so that you can generalize beyond them. The coincidence of the location of this lab, which studies exactly this kind of virus, and the outbreak itself. And that's a problem, right? So that means that it's reasonable to look at that and think that, well, it could have escaped from a lab there. That's the first conclusion. And that it has to be demonstrated in some sense that it didn't. And then, so that's a problem. And I can't see how that's anything but an incontrovertible problem. The mere fact that that lab is there and that it does research on those types of viruses and that that's where the outbreak was doesn't prove that it originated in the lab, but it certainly makes that a plausible hypothesis. But then you add this additional twist, which is, I think, more complicated for people to understand. And you detail this out, you provide some detail for this in the book, that this virus is somewhat remarkably well adapted to human beings. Now, there are literally trillions and trillions of different forms of viruses. And so obviously most of them aren't particularly well adapted to human beings, because otherwise we would have trillions of viruses producing pandemics through all the time. So it's generally the case that viruses are not well adapted to transmission in human beings, and that's true for the overwhelming majority of viruses. And so the fact of this human adaptation, or adaptation to human transmission, is something of signal importance. And so maybe you could walk me and everyone else listening through why a typical virus isn't adapted to human transmission and what it means that one is and how that develops.
Yes. The normal pattern when a virus first emerges into the human species is for it to be very difficult for the virus to spread human to human. It can infect someone, it can even possibly kill someone, but they're not very good at passing it on to people. The virus is not really very good at transmitting between members of this new host. Now, if enough time goes by with enough infections happening, then eventually it will get good at it. And that's what was starting to happen with SARS in 2003. It first infected people in the fall of 2002. By the spring of 2003, you were starting to see chains of transmission from person to person. And the reason for this is that the virus has to evolve. It has to change its genetic code in such a way that it can better fit the receptors on the cells of humans as opposed to the receptors on the cells of bats, or in the case of SARS, the intermediate host, which was a palm civet.
So we should point out that the problem of transmission that a virus has to solve is no different from the problem of the virus determining in some sense or evolving so that it can live. Transmission in a virus is the same thing as propagation in the environment. And so this is a very knotty problem, KNOTTY, for a virus to solve. It's not simple at all because it has to not kill the host too quickly. And it has to be able to replicate within an individual, but then it not only does it have to manage those things, which is very difficult, it also has to figure out how to transmit itself with some degree of effectiveness. And thank God most viruses can't solve that problem. So this is a very thorny problem. And maybe you can outline for everyone how viruses do solve that problem and why most don't, and why the fact of human adaptation to humans is so important.
Yeah. So just to give you a good example, bird flu, which is a big problem at the moment in poultry flocks and wild birds, is not very good at infecting human beings. People can catch it. People have even died of it. If you're exposed to a huge dose by working in a poultry farm, you can get sick and you'll probably die, but you probably won't give it to anybody else. We've not seen any human-to-human transmission of that virus. This virus was good at transmitting between human beings from the word go, from the November or December of 2019. And the reason for that, we now know in molecular terms, it has a so-called receptor binding domain on its spike gene, which is well adapted to the human ACE2 receptor on our cells. Not very well adapted to other species receptors, which is intriguing.
So it's a specific human adaptation.
Well, not specific. It's not completely specific. It's quite good at lots of animals, but it's really good at us. Okay. And the other feature that it has that is very striking and very surprising is that it has something called a furin cleavage site. Now, this is a small section of the spike gene that is coded by about 12 letters of RNA genetic code, which has been inserted into it, compared with all its close relatives. And that gives it the ability to use a human enzyme called furin that is in all our, many of our cells, in order to reconfigure the virus as it leaves the cell to make it, to as it were, prime it to attack another cell. So it's actually using one of our enzymes to spread in the body. This enables it to infect more tissues in the body and to multiply much more effectively. So it's the reason we're having a pandemic, is the furin cleavage site. If this virus didn't have that, it would probably have been pretty easy to control in the early months.
You said something very interesting, and this is another piece of the puzzle, I presume, is that compared to other viruses of its type, it has this interesting furin cleavage site. I've got that right. And you said that it had been inserted, and that's an anomalous statement, let's say. So there's something about this particular virus that sets it out against other viruses of its type, and it's this particular ability to use a human enzyme, and you described that as inserted. And so I presume that could be inserted as a consequence, hypothetically, of natural selection processes, or that there's other alternative explanations. So what do you see when you look at that?
What I mean by inserted here is that it's an extra piece of genetic information. We can look at 20 or 30 other very similar viruses, and we can line their genomes up and match them up against this one. And the furin cleavage site is not an altered bit; it's an added bit. It's an extra chunk that's been added in the middle of the spike gene.
So how do you differentiate between added and different? I presume that's a consequence of degree of divergence. So you could imagine that there's a set of viruses that are genetically and evolutionarily similar, so they stem from a similar source, and they have a predictable pattern of variation. And you're saying that this is an anomaly that exists outside that.
Well, you line them up. You align the genetic codes of the viruses, and in this particular part of the spike gene, they all align up quite well. You know, the sequence is mostly the same. You can tell it's been added because you can align the genes of the spike genes of other viruses alongside each other and see that this is an extra piece of RNA, not a changed piece of RNA. It's about 12 letters long. And this spells out a sequence that allows, it's called a furin cleavage site, and it allows the virus to use a human enzyme called furin to spread from cell to cell, from tissue to tissue, and effectively makes the virus much more dangerous and much more transmissible. It's the reason we're having a pandemic. If the virus didn't have this furin cleavage site, we'd probably have been able to get it under control very easily early in the pandemic. Now, what's interesting is that a number of Western virologists, when they first saw the sequence of this virus, said, whoa, it's got a furin cleavage site in. That's very unusual for a SARS-like virus. In fact, it's unique. We've never seen one with this before. There are other coronaviruses with furin cleavage sites, but not SARS-like coronaviruses. And they said, I'm afraid that suggests it might have been engineered.